After our survey visit on the J-3K planet, or Mala planet, as a chosen group of scientists, we are proudly to present all the information that we collected and analyzed based on our observation on this planet. Let's begin with the condition.
So we start with solar system. Mala planet orbits the sun at quite close to the sun. The distance is about 100 million kilometers. It takes approximately 18 hours on Earth for Mala to complete one day. And next is atmosphere. Mala's planet surface experience extreme temperature variation during the day. Temperature can rise up to around 7 to 130 degrees and has an extremely thick and dense atmosphere, or almost sealed, and covered by greenhouse gas, which means it lacks the protective layer that helps regulate temperature on Earth. As a result, the atmospheric pressure on Mala planet is much higher than Earth. The surface pressure on Mala planet is about 2 times that of Earth. Sea level pressure. To provide a specific value, the average surface pressure on Mala planet is approximately 2 bars, whereas 1 bar is equivalent to atmospheric pressure at sea level on Earth. But the gravity is still be a standard gravity as on Earth, which is 9.8. Now we're moving to a continent. The land on this planet is both an island and continent, and it is surrounded by water unlike other planets. It doesn't share land borders with other continents. Next is biome. Mala planet only have 2 biomes. A tropical season forest, also known as a tropical dry forest, is a type of biome or ecosystem found in regions with a distinct wet and dry season. Next is savanna or tropical savanna. It's a biome characterized by a combination of grassland and scattered trees, and it is found in both tropical and subtropical regions. Savanna are known for their distinct seasonal rainfall pattern with wet and dry season. Now let's move to organism.
Although planet conditions are harsh, but with the abundance in the material, many organisms are thriving as well as diverse in its own way, both the aquatic and the land organisms. Many aquatic get their nutrition from the underwater volcano. For the land, most plants have deep roots to extract nutrition. Most produce the capling with dense in calories but high in concentration of capsaicin. So many of the animals become carnivore, but some unique traits allow them to survive using the capling, such as the shooting bee. These creatures have unique genetic traits allow them to consume capling without problem. Now we're gonna move on to the genetic approach.
So, the way we approach to our study is through the QTL analysis or qualitative trait locus, as it provides understanding in the genetic basic of these traits such can be applied to various fields. In this analysis consists of various procedures which will be start with population mapping.
After we landing, we set the research station and observe organisms on the planet. And this is what we observe about the organism. We observe this organism about their mating duration, type of embryo, number of offspring per cycle, and gestational period. They have 1 to 2 hours mating duration. They produce around 10 offspring per cycle, take full 5 months to hold babies, and they are opportunistic breeder, which means they have capable of breeding at any time or becoming fertile in a short period of time. Studying quantitative trait loci often benefit from a large number of heterozygous individuals because it allows for increased genetic diversity and a better chance of identifying genetic factors influencing quantitative traits. Thus, we have planned to captivate breed for 100 generations based on our population mapping. First, we do double haploid. Double haploid technique aims to generate pure inbreed line for basic research and as commercial cultivars. The double haploid technique first generates haploid organism and followed by chromosome doubling, which can be separate, overlap, and can see variety in a short time. Then we use backcross to identify if the parent generation that we breed is homozygous or not. If the recurrent parent is an elite genotype, at the end of the backcross program, an elite genotype is recovered as new recombination results. The elite combination is not lost. If we cross F1 and P2, percentage of offspring must be 50% homozygous recessive to prove that the P2 is pure breeding or homozygous recessive. On the other hand, if we cross F1 and P1, percentage of offspring must not contain homozygous recessive to prove that P1 is pure breeding of homozygous dominant. If the result of backcrossing is not pure blood breeding, go back to double haploid process until you find the right parent. Once we already found the suitable parents, we do captive breeding for 100 generations. No migration is allowed. We randomly pick every single one of every generation to collect saliva sample. After that, I send the sample of saliva to the lab team. Let them extract casein, which can bring capsaicin from free nerve ending. When the extraction is done, we create the table that shows the level of capsaicin tolerance based on amount of casein in saliva.
Now we're going to moving on to the PCR and DNA extraction. So the reason why we choose saliva as DNA template for PCR is based on our observation during the visits. Some of them didn't consume the plant as they contain capsaicin, which can be concluded that the non-plant eater might lack the ability to build specific enzyme for capsaicin, like casein for instance. So to understand that, we're collecting saliva from pure breed to extract out the DNA, then isolate the DNA and measure the concentration of the extracted DNA. After obtaining the template, we then proceed to the PCR method. Here's our PCR mixer. Keep in mind that in this experiment, there are 5 markers to test for. Each marker consists of 100 DNA templates, 2 templates from the parents, and each negative and positive, making the solution on each marker to be 104 solution, hence the overall to be 520 solution. Now moving on to the PCR program. Here's our program. As you can see, the holding process is done until the gel is perfectly formed and ready. However, consider storing at -20 degrees Celsius or -80 degrees Celsius can be a way to avoid degradation over time. Now, after the gel is ready, we're gonna using the gel to visualize the PCR product to confirm the presence and size of the amplified DNA fragment. After that, we will get the outcome from the gel, which will look like this. Then, we're using the gel results to make the marker scoring and coding figures as shown. Now, we're gonna utilize the figure to aid in the QTL analysis and linkage map.
For the QTL analysis, after we got marker scoring and coding, we use QTL analysis method. Based on the results, marker S is linked to a QTL because the significant difference between means, and for the other markers are linked to QTLs because there are no significant difference between means. The closer the marker is to QTL, the lower chance for recombination between marker and QTL. Next is our linkage map. After we found that marker S is linked to QTL, so we can detect the locus of AS7S7 gene in the linkage map, which is located in the chromosome number 2.
So, thank you for joining our celestial journey. May our gaze upon this wondrous planet ignite an unwavering commitment to safeguard its splendor for all time. May the force be with you.